Rupinder Kaur from Department of Biochemistry, AIMS, New Delhi.
So very good evening to everyone. Uh, I'll be presenting an updated protocol to establish single cell originated sprite culture. So this is the poster view, and I've taken the liberty to dissect into different slides. So in the current research scenario, the most promising areas expected to improve the success rate during drug development, uh, especially for cancer, is the availability of new preclinical models that better recapitulate the in vivo biology and tumor microenvironment. So tumor microenvironment, if needed to be studied, ex vivo 3D cultures are thought to be best suited models for the purpose because they form a perfect bridge between in vitro 2D monolayers and in vivo animals, which are relatively difficult to handle. So in our lab, we have established an economical protocol to make these single cell originated spheroids. And for that purpose, we had used 5 different cell lines as given here. And we took the advantage of the fact that when these cell lines are grown in 2D monolayer cultures, they have a very low population of cancer stem cell-like cells, which have very low adherence to the culture flask surface. And such that even if trypsin is administered in very low concentrations, say 0.05%, they easily detach from the surface and can be made a suspension culture from that. So these trypsin sensitive cells are enriched from 2D monolayers by incubating them for 2 to 3 minutes at 37 degrees Celsius. And these trypsin sensitive cells can be further subcultured to enrich their population. Mostly 3 subcultures are sufficient. And these trypsin sensitive cells, when enriched, can be plated onto a low adherent surface to make cancer spheroids. So to serve the purpose of getting a low adherent surface, we used a hydrogel such as agarose, and we optimized its concentration, and it was found that 0.5% of agarose worked well for the purpose. And after 5 days of seeding the cells onto this low adherent surface, we got very good number of single cell originated spheroid cultures, which were further analyzed for the presence of stem cell markers like CD24, CD44, CD133. The spheroids obtained were also analyzed for their proliferation, as their mitochondrial potential assay and the cellular health assay. These are the results which we obtained. As you can see in figure 2a, these are the 4x images of Cal33 obtained spheroids, and there is a comparison between 2D cells and the trypsinized cells. So in figure B, there is a time-lapse image of 24 hours, 40 years on subsequent time, where cells first come together as clusters and they form a compact spheroid later at 120 hours. In next figure, adjacent figure, you can see the immunocytochemistry, where these spheroids from 2 different cell lines. In figure A, it's Cal33, which is labeled with cancer stem cell-like markers like CD44 and CD24. And you can see a merged image here. In figure 3A and 3B, this depicts the immunofluorescence images of 2 different cell lines, Cal33 and MDMB231. As you can see that there are significant fluorescence of CD44 and CD24 markers, which are cancer stem cell-like markers. In figure 3C, you can see the mitospy dye, which shows that the spheroid cells inside the spheroids are metabolically active. And in figure D, you can see that there are actin filaments, which are red in color in the third figure, which shows that the cells of the spheroids are very densely packed through cellular cytoskeleton elements such as actin. So this is the data which shows the flow cytometric analysis of 2D cells as well as 3D cells for the presence of cancer stem cell-like markers. So in figure A, you can see that the 2D cells represents very low percentage expression of these cancer stem cell-like markers, which is from 6 to 14% for different markers, and it increased significantly to 80 to 90% in case of trypsin-sensitive cells. And when these trypsin-sensitive cells were further plated on to low adherent surface, it was found that the percentage expression is lesser than trypsin-sensitive cells, but it was significantly higher as compared to the 2D cells. And it was observed that the expression remained constant up until day 8, and which reduced further on for subsequent days. So this are the figure which shows the cell proliferation data of 2D cells versus 3D cells. Figure A shows 2D cells and B shows 3D cells, and it can 90% cells remained undivided. That is, they entered the stage of quiescence, which is a characteristic of cancer stem cells, which are present inside the spheroid core. So the adjacent figure shows the mitochondrial membrane potential of cells, 2D cells versus 3D, as mentioned. So this mitochondrial membrane potential assay is the depiction of the cellular activity, basically. So it was observed that 90% of the cell population in 2D culture, they showed high mitochondrial membrane potential, whereas only 76% of the cells in 3D culture showed such high membrane potential. That means that the cells inside the core have lesser mitochondrial membrane potential and hence cellular activity as compared to the 2D cells. So this last figure shows the cellular health of the cells inside the core. As you can see in the fluorescence image, B, 3D cultures, you can see that there's very little inflorescence, very low intensity of the dye inside the core, which depicts that the cells inside the core have started to deteriorate in health, and they will subsequently die because of the deprivation of nutrients, as well as the generation of hypoxic conditions because of lesser oxygen in the core. So the presented data concludes the cancer stem cells can be isolated very effectively using trypsin-sensitive methodology, and the isolated cells have a significantly higher expression of cancer stem cell-like markers, and which is very much higher as compared to the 2D monolayer cultures. Also, it was observed that as the spheroid size increases, the cellular health and cellular viability decreases inside the core, which is well accepted as there is a lack of nutrition and generation of hypoxic conditions inside the core. While in vitro, these hypoxic and nutrition deficient conditions leads to necrotic core as seen in the data. In vivo, much more resistant cancer cells are there in such necrotic cores, which escapes this hostile environment, and later they cause metastasis in cancer patients. So these are the references. Thank you all. Any questions?